The potential for lab-grown biological tissues could be unlocked through the use of aligned peptide noodles. Greetings, everyone. Today's video focuses on an exciting development in the field of research. Scientists have successfully engineered aligned peptide noodles that imitate the intricate structures of muscle and nerve tissue, which are essential for proper functioning. These aligned peptide nanofibers have proven to be highly effective in guiding the growth of cells. However, it is important to note that excessive alignment can actually impede cell alignment, underscoring the significance of nanoscale interactions between cells and materials. This newfound understanding has the potential to greatly enhance tissue engineering strategies. The study received support from various grants and involved collaboration between researchers from Rice University and the University of Houston. Without any further delay, let us dive into our discussion. Professor Jeffrey Hartgerink, along with his research team in the fields of chemistry and bioengineering, has successfully created peptide-based hydrogels that imitate the organized structure found in muscle and nerve tissues. The alignment of these tissues is crucial for their proper functioning, but replicating this characteristic in a laboratory setting is a complex task that involves the precise arrangement of individual cells. Over the course of a decade, the team has dedicated their efforts to the creation of multi-domain peptides, MDPs, that possess the unique ability to autonomously arrange themselves into nanofibers. These nanofibers bear a striking resemblance to the natural fibrous proteins present within the human body, akin to a minuscule spider web. The researchers have recently conducted a study that has been published online and highlighted on the cover of the journal ACS Nano. In this study, they have unveiled a fresh approach to produce aligned MDP nanofiber noodles. Initially, they dissolved the peptides in water and subsequently extruded them into a solution containing salt. This innovative technique allowed them to generate aligned peptide nanofibers, resembling tiny twisted ropes that are smaller than a cell. Furthermore, by elevating the concentration of ions or salt in the solution and repeating the procedure, they were able to achieve even greater alignment of the nanofibers. Lead author Adam Farshid, who recently achieved his PhD in bioengineering from Rice, clarified that their discoveries showcase the capability of their technique to generate peptide nanofibers that skillfully direct cell growth in a specific orientation. He emphasized the significance of this advancement in the development of functional biological tissues for the field of regenerative medicine. A significant revelation from the research was an unforeseen revelation. If the alignment of the peptide nanofibers was excessively robust, the cells ceased to align. Upon closer examination, it became apparent that the cells required the ability to exert a pull on the peptide nanofibers in order to perceive the alignment. In instances where the nanofibers possessed excessive rigidity, the cells were incapable of generating this force, resulting in their inability to arrange themselves in the intended configuration. Hartgerink expressed that this understanding of cellular behavior has the potential to extend its influence to the fields of tissue engineering and biomaterial design. By comprehending the intricate interactions between cells and nanoscale materials, there is a possibility of developing more efficient approaches to constructing tissues. Among the co-authors of the study from Rice University are Tracy Yu and Carson Cole, who both hold PhD degrees in the chemistry department. Joseph Swain, a graduate student, and Adam Thomas, an undergraduate researcher, also contributed to the study. Additionally, Jonathan McCool, an undergraduate researcher in bioengineering, Eric Garcia Huitron, a graduate student, and Professor K. Jane Grand Allen were co-authors on the study. The team of researchers from the University of Houston included Christian Zavalos Delgado, a PhD student, Sajede Saidifard, and Manmohan Singh, both research assistants, and Kirill Larin, an engineering professor. Partial support for this undertaking was provided by the National Institutes of Health through grants R01D02-1798, R01IY022362, R01HD095520, and R01IY030063, as well as the National Science Foundation Grant 21291122. Additionally, funding was received from the National Science Foundation Graduate Research Fellowship Program and the Welch Foundation, C2141. It is important to note that the authors bear sole responsibility for the content of this press release, and the viewpoints expressed herein may not necessarily reflect those of the funding entities. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.